Hello friends, today we are going to discuss one of the important technique in molecular biology about gene transfer methods in recombinant DNA technology. Gene transfer methods in recombinant DNA technology. Before we go into the topic, what is the significance of gene transfer? In the last class we have discussed about the basic protocol of genetic engineering or recombinant DNA technology. First we have to generate the DNA fragments, then selection of the desired DNA fragment, what we call a specific gene of interest, then incorporation of the specific gene into a plasmid, say for example a vector, what we call creation of chimeric vector, then introduction into the host cell, that means say that what we call cloning factories. Host cell may be an Escherichia coli. We have discussed at length about different host cells. That means in animal cell, for example, a insect cell, or in oocyte, okay? And then multiplication selection of specific clone containing the desired recombinant DNA molecule. Finally, the synthesis of the end product, that means the protein, okay? Here the third step, introduction of recombinant plasmid, or say chimeric DNA, or simply what we call a chimeric vector, into the host cell, what we call gene transfer into the specific host cell is very important, because I told you what is the significance, the crux point of the success of recombinant DNA technology or a gene cloning technology depends on this very essential step, that means gene transfer. There are four different ways generally, say transformation, and conjugation, electroporation, then liposome mediated gene transfer. These are the indirect ways. But there is one more, what we call direct transfer of DNA. Direct transfer. Say it is by microinjection and particle bombardment. These are the different ways of gene transfer that are commonly or generally used in recombinant technology. That means, so that the same will be introduced into the host cell, maybe an Escherichia coli for instance, or maybe oocyte, or even a protoplast. Okay, then here the transformation, the conjugation, electroporation, liposome mediated gene transfer, lipofaction, then the direct transfer method. There are two ways of directly transferring the recombinant DNA into the nucleus of the concerned host cell. It is through microinjection, and next one, what is the most advanced, what called particle bombardment. Okay, we'll discuss one by one. With respect to the transformation, it is one of the most simple and reasonably good method. Okay, we will briefly describe how the foreign DNA, how the recombinant vector get inserted. So this is what introduction into the host cell. Say there are specific requirements. Say for instance, 0 to 5 degree centigrade. That means what we call ice cold CaCl2 treatment plus heat shock treatment, that means for 37 to 45 degrees centigrade for about 90 seconds. To introduce the foreign DNA into host cell, we have to maintain this ambience. Without which it is impossible. Say ice cold CaCl2 the rate of somewhere between 0 to 5 degrees centigrade, followed by heat shock so 37 to 45 degrees centigrade, so that these enable the foreign DNA to introduce into the species of body. The most important and the significance point of transformation is it is reasonably better than other techniques. Say what we call transformation frequency, simply called transformation frequency. Say it is 1 in 1000 cells. That is what 10 to the power of minus 3 cells. You know what is transformation frequency? The proportion of the cell or cells which has been transferred in a group of cells, or the percentage of specific cells in a group of cells that has been transferred. This is transformation principle, and it is reasonably better in case of transformation if we use this method for gene transfer when compared to other methods. Okay, the next one is conjugation. We have to speak about this at length. Now, what is conjugation? Conjugation is a natural microbial recombination process. I'm not going to the details of the basics. Say, for example, we knew that, say, there is a F plus, or that means F plus versus F minus. F plus means contains F factor, what is called extra chromosomal DNA. F minus doesn't contain. Here F plus is the donor, whereas F minus is the recipient. The duplicate copy of F plus will transfer into the F minus through cytoplasmic bridges, so that the F minus becomes F plus. That means microbial recombination. The same principle has been used in gene transfer method. Say we got a recombinant plasmid, so as to enable to transfer into the far, that means host cell. The same concept, say a formation of cytoplasmic bridges between the two live bacteria, say the F plus and F minus, just like F plus, so that you can transfer through cytoplasmic bridges into the recipient cell. It is one of the most convenient method. Okay, say for example how it ends. This is called F plus. And these are called cytoplasmic bridges. This is normal chromosome. Okay, this is F plus. This is F factor. This is normal chromosomes. This is F minus, and this is cytoplasmic bridge. And the next step, the duplicate this has to transfer here. What we call through conjugation, it has to get transferred. Here. Then finally we get what we want. That means both become F plus. This F plus, F factor, what we call. This become F plus and F plus. Okay, this is how the issue ends. You know, in the initial conditions, the F1 doesn't have this extra chromosomal entity, extra chromosomal DNA, what we call F factor. Later, through the formation of cytoplasmic bridges, it has been transferred into the recipient. Okay, that means here it is a donor and this is a recipient. This is a recipient. Okay. Finally, both become F plus. That means it contains extra chromosome DNA. Based on the same principle, the recombinant plasmid, say, that has been transferred. In place of F factor, the recombinant plasmid has been transferred. The most important point is the F factor. That means may not, generally, rarely it integrates into the normal chromosome, but in general it remains free. For example, they live as separate entities. Separate entities. Most of the, what we call rarest of the rare cases, this F factor may integrate into the normal chromosome. The same concept here. That means not all plasmid, that means bacterial cells have this procedure, but some may have this. This is very important. Okay, so and by employing this technique, the recombinant plasmid has been transferred into the concerned host cells and allowing it to multiplication. Then we have to identify the specific clone which contains the recombinant DNA molecule, then allowing it for, that means what we call synthesis or translation, simply what we call the end product. Say we want an insulin gene, then the end product is insulin. So we have to allow it through proper molecular machinery. This is how the issue with respect to conjugation. Okay. Then the most important, what we call electroporation. This is something like applying of physics techniques. We knew that kinetic energy K equal to half mv square. The same concept we have to apply here. In general, the cell membranes, they are not static. They are mobile. We have discussed in our previous classes about cell membrane and different membrane transport system. Here, the phospholipid bilayer is a mobile, what we call fluid mosaic model now. The fluid with respect to lipid domain, whereas mosaic with respect to protein domain. And this mobile nature of phospholipid enables it to have potential kinetic energy. And when this kinetic energy get accentuation through, say, high voltage electric shock or high voltage electric field, and this energy K become enhanced. This enhancement has been materialized in increased electropermeability. That means the membrane permeability of the plasma membrane has been increased, which has been reflected in the formation of electropores, what we call electroporation. These are temporary pores, they are not permanent. For a while they will form, and through these pores, the foreign DNA, what we call the recombinant DNA, has to be, it get inserted inside the cell and eventually get inside the nucleus. This is the basic technique in electroporation. Simply the subject the cell to high voltage cell, to high voltage electric field. Then the cell membrane, it rises its kinetic energy, that K equal to half mv square, and it rises in turn leads to increased permeability, what we call electromediated membrane permeability. Is very important. That means in general they fuse. The same concept here. That means inclusion of exogenous DNA through these pores is a basic touch point for electroporation. How this ends. For example, electroporation. Say this is a cell culture. And this is a recombinant plasmid, and this is our target gene, and this is marker gene of interest. This is a marker gene. Then combine the same and place it in a cuvite, a specially designed cuvite. That means it contains a mixture of both the cell culture, that means the cells, and the rDNA. For example, say plasmid, the mixture. Okay, subject the mixture to high field, that means high voltage electric field. To high voltage electric field. High voltage electric field. Then what happens? here the recombinant DNA will get inside the cell. So we will magnify. For example, this is a cell and this is the nucleus. You know how? Say I wanted to magnify here. Say through formation of some pore, temporary pores, this recombinant plasmid, has got inside. This is electric pores, and this is the R plasmid. This is just I would like to get magnified so that ultimately, and this is the target cell and its nucleus, and this nucleus contains recombinant plasmid. This is a basic, with the basic principle, K equal to half mv square. This is kinetic energy, Generally, I told you already. When we apply, because of the very nature of, that means the very mobile nature of the phospholipid nature of cell membrane, this has been done. Say apply high voltage electric pulse and raise kinetic energy, it increases membrane permeability, what we call electric mediated membrane permeability. This leads to this, and this is because of mobile nature. There is a link, okay? Then that leads to electroporation, the anti-scenario. This is the basic technique employing in electroporation. That means you have to understand certain physics, that's it. Then the most next important way for gene transfer is liposome mediated gene transfer. Liposome mediated gene transfer. Liposome mediated gene transfer. Now how it ends? We knew that what are liposomes? They are circular lipid structures. That means there will be a phospholipid bilayer, and inside there will be an aqueous content, and this aqueous content contains a DNA. How they will facilitate what we call lipofaction. That means liposome mediated gene transfer is called lipofaction. Okay. How they facilitates the transfer of, that means the introduction of recombinant DNA into the target cell. We all knew that there is the nature of liposome. They fuse with the cell membranes. For example, this is a target cell and this is liposome. They will fuse the target cell, and after they fuse, the inside content of the liposomes get discharged inside that means just like transport system inside the cell, and they eventually goes to the nucleus. That means membrane fusion mediated. That means liposome, then fusion with membrane, then simply what we call discharge. This is the basic mechanism of liposome mediated. And for liposome, there is one more important and interesting thing. DNAs are negatively charged molecules, but liposomes are positively charged molecules. This is very important. The positive liposomes and negative DNA molecules. This is very important for the formation of liposome DNA complex. Electrophysics, simply what we call. This positive negativity plays an important, positive liposomes and negative DNA plays an important role in formation of complex, then eventually cell membrane fusion, then discharge. How it ends? For example, say this is a lipid solution. Lipid solution plus, say, is it the appropriate, the DNA followed by creation of a liposome. Now just they are considered as a two membrane, It is a lipid bilayer. Because of time constraint, I am just speeding up It means that it is a lipid bilayer, okay? A lipid bilayer. Then inside the target DNA molecules. Then the next step is one of the normal. This is a host cell. Then these they fuse with the, this is liposome, and this is host cell. Or what we call fusion. Then they discharge the contents inside the cell. Eventually the DNA make it to the nucleus, or DNA inside the cell. This is a basic mechanism of liposome mediated gene transfer, we call lipofaction. Simple. The positively charged lipid molecules get attracted to the negative charge, and this enables the formation of liposome DNA complex. And after that means liposome has been circled, that means DNA has been encapsulated in the liposome, what we call encapsulation of DNA. That means it's a simple concept. Encapsulation of DNA by lipid structure. That means a circular lipid structure. Then this lipid component binds and adheres and fuses with the target host cell. Then fusion. Then discharging the inside the DNA inside the cell, and eventually to the nucleus, make it to the nucleus. This is how the issue ends with respect to liposome mediated gene transfer. Okay. Then the last one, which is a bit advanced, when we at some other occasion will speak at length about direct method of gene transfer. At some other question we'll speak, because it requires lot of explanation and discussion. Specifically, it has to be focused. Say for now we will briefly discuss, okay, what we call direct DNA transfer. How? Simply it is by micro injection. Then particle bombardment. Because through these two techniques, DNA, instead of what we call mediators, electroporation is a mediation, what we call electroporation, electric mediated permeability, or liposome mediated, or conjugation driven, or transformation. These are called indirect ways. We require some facilitator, but here just include the recombinant DNA directly inside the nucleus of the target host cell. This is very important. Simply it will be done by a micro injection technique and proton bombardment. I would like to give one more thing, that means point on this topic. We all knew that there will be what we call cloning strategies, and followed by, and which component will be used in cloning. Generally, the genome, that means which is nothing but the entire DNA content of a cell or even entire organism, generally used for the cloning experiments. But scientists has been shifting from DNA, that means genome, to mRNA. Why? Because genome contains unwanted things like repeated sequences, introns which are non-coding sequences, and regulatory components. But mRNA is called extract form of the gene. That means the full-fledged what exactly the gene wants to say has been present in mRNA. Because in mRNA there is a process we have discussed in our previous class, what we call post-transcriptional modification. That means splicing. The introns will be removed by spliceosomes, two SNRPs. Okay. Then exons will be joined together, which is complete compact gene structure. Information has been present in mRNA. Therefore, the same has been used in different cloning experiments, because there are several other advantages. Say selection and isolation of mRNA is very easy, and synthetic protein synthesis, what we call synthesis of recombinant DNA proteins, is also very easy from mRNA. So because of this, in cloning experiment, mRNA has been given the prima facie importance than the genome, except if our experiment is focusing on gene experimental regulation things, the rest of the things are completely depending on mRNA. Okay, this is about today's class. In next class we can discuss some other topic. Okay, you have to depend on some reference and it's a basic book for consolidation of your knowledge. Okay, thanks for watching.